everyone, welcome to Agri Addict YouTube channel. I'm your tutor Sakhi. Today we'll be discussing the third part of crop physiology lecture series. In this, we'll discuss the photosynthesis in plants. Okay, let's start with the lecture. This is my short intro. I have done my undergraduation from JNKV Jabalpur. Currently, I am pursuing masters at IRI Hyderabad Hub FREDA Department of Agronomy. I have secured All India Rank 12th in ICR PG 2024 examination. So let's start with the lecture. So first, what is photosynthesis? So photosynthesis is a physicochemical or photobiochemical process. Why we are saying photo? Because it is requiring light. Okay, photobiochemical process in which the organic compounds like carbohydrates are synthesized from inorganic raw material, that is water and carbon dioxide, in the presence of light, that is sunlight, and pigments. Pigments are chlorophyll. Main pigment is chlorophyll. And here O2 is evolved as a byproduct or one of the net products. Okay. So let's see. So photosynthesis is a biochemical process. It is not a single step reaction. This is what we are seeing, that 6CO2 plus 12H2O in presence of light and chlorophyll. It, it is synthesizing glucose plus O2 is getting released plus 6H2O. This is not a single step process what we are showing. It is a multi-step process. Okay. It is an anabolic process, means something is getting synthesized. What is getting synthesized here? Glucose is getting synthesized. Okay. So there are two kinds of process. One is catabolic, another is anabolic. Catabolic, as it says, is we are cutting down something, we are breaking. What we are doing is here complex substances are getting converted into simpler ones. And in anabolic, something is getting synthesized. So photosynthesis is an anabolic process and it is an endergonic reaction, means it is using energy. What kind of energy? Here the energy is being taken by sunlight. Okay. Next. So photosynthesis is a redox reaction. Why we are saying it is a redox? Because reduction as well as oxidation is taking place. What is getting reduced? Carbon dioxide is getting reduced and oxidation of water. Oxidation of water, why we are telling? Because as it provides hydrogen ion and an electron in the light reaction. Whereas there is reduction of carbon dioxide. Why we are saying carbon dioxide is getting reduced? Because it is accepting that H plus and electron which is released in dark reaction. So this reduction of carbon dioxide takes place in dark reaction, which is also known as biosynthetic phase. Here light energy is converted into chemical energy by photosynthesis. Okay. Next. So let's start with the experiments. So first is Mole's half-leaf experiment. In this, what Mole's did is he took one plant, a pot plant. In that, one part of half part of that leaf he covered, kept inside this box, where which is in that one KOH soap cotton was kept. Okay, this KOH has this property to absorb carbon dioxide. So later on, he analyzed this leaf. So the part of leaf which was inside this structure, it tested negative to starch. Means that part of leaf didn't synthesize starch. Whereas the part of leaf which was outside of that structure, this tested positive for the starch. It gave blue-black color. Okay, so it symbolized that there is a presence of starch, means starch was synthesized in that part of leaf. So what he made conclusion is that CO2 is required for sugar synthesis. For sugar to be synthesized during this photosynthesis, CO2 is an important component for this. Next comes the inverted bell jar experiment. This experiment was given by Joseph Priestley. Okay, this experiment was done by Joseph Priestley. So what he did in one bell jar, this experiment, he put a lighted candle in it. Okay, after some time he saw that this candle was not lighted. Okay, in the second scenario, what he did, he put mice, alive mice, inside this bell jar, and he found after some time, he found that the mice was dead. In the third scenario, he put lighted candle, alive mice, and a mint plant in that one bell jar. After some times, he found that all the three, that is, the candle was lighted, that mice was alive, as in the previous scenario, and mint was also healthy. So, what conclusion he made is that plants store to the air whatever the breathing animals and burning candles removes. He told that whatever the things they are breathing out, like candle and mice is breathing out, it taken by this mint plant or this plant, and whatever the mint plant is releasing is taken out by this candle and mice. So that's why all the three were healthy inside. Okay. So he said that some foul air is being released by candle or mice, which later on found to be carbon dioxide, whereas something good air this plant is releasing, which is consumed by this candle and mice, which is found later on found to be oxygen. Okay, this is how the oxygen and carbon dioxide is known to us, that it is an important component of photosynthesis. So let's move to the next slide. Now comes the Jan engine house experiment. This experiment tells us the importance of sunlight in photosynthesis. Okay, here we got to know that sunlight is important for photosynthesis to be taken place. Okay, so first, in the first scenario, what he did, he took one hydrilla plant, which is a water loving plant, okay, kept in water. And in the second scenario, one scenario, it is in presence of sunlight. In the another scenario, he removed the sunlight, okay, it was placed in dark condition. So he found out that this at the corners of this hydrilla leaves, he found some bubbles. Okay, that bubble he recognized to oxygen bubbles. Later on, he found out that it is oxygen bubble. So Indian house in an elegant experiment with an aquatic plant. This aquatic plant is this hydrilla, showed that in bright sunlight, small bubbles were formed around the green plants, while in dark, they did not. Later, he identified these bubbles to be oxygen. Hence, he showed that it is only the green part of plant that could release oxygen. First takeout from this experiment is that it is the only green part of plant that could release oxygen. Second conclusion is for the release of O2, sunlight is required by green plant. This is an important conclusion. Jan Ingenhouse's experiment tells us that sunlight is an important and it is required for green plants to release O2. Let's move to the next slide. Now comes the Julius Swan sac. He told that special bodies contain special pigments in which glucose is formed. And these special bodies later on we call that chloroplast, whereas the special pigments are chlorophylls. Okay. Green substance in plant that is we call now chlorophyll is located in special bodies which is called as chloroplast within plant cell. He found that green parts in plant is where glucose is made. Green part of the plant where glucose is formed, and that the glucose is usually stored as starch. That's what he said. Okay. Next is Engelmann's experiment. So what he did, Engelmann in his experiment used Cladophora algae. It is an algae. He took a white light, he passed it into the prism, and this prism helped that light to get split into different wavelength, that is Vapeur. You might be knowing this. Violet, indigo, blue, green, yellow, orange, red. They are the visible light range. Okay. So for 400 nanometer to 700 nanometer. So he found this cladophora algae, the aerobic bacterial growth is more in this area, which is blue and red. And aerobic bacteria require what? Oxygen. It means more production is of oxygen is taking place in this blue region and this red region. So later on, the conclusion he made was red and blue light wavelength zone has maximum O2 release, and hence it we can say that it is the area which is having maximum photosynthesis, or the rate of photosynthesis in this area is more. Okay, that's what Engelmann said in his experiment. He used cladophora algae for his experiment, and he found out that aerobic bacterial growth is more near to this blue wavelength and red wavelength. Okay, so let's move to the next slide. Now comes the one Neil. Cornelius von Neil, based on his studies on purple and green bacteria, he was doing study on purple and green bacteria. He demonstrated that photosynthesis is essentially a light dependent reaction. That's what Jan Ingenhouse previously told, in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. What we study? See, CO2 plus H2O gives us CH2O, that is a carbohydrate. It is a basic formula of carbohydrate. So here what is he's telling is hydrogen from a suitable oxidizable compound. Here suitable oxidizable compound what we study is water. It reduces carbon dioxide. Carbon dioxide is getting reduced in a carbohydrate to carbohydrates. In green plants, H2O is a hydrogen donor. In case of green plant, H2O act as a hydrogen donor and is oxidized to oxygen. Here, oxygen is getting released. Some organisms do not release O2 during photosynthesis. Why? When H2S instead of hydrogen is the hydrogen donor for purple and green sulfur bacteria. These purple and green sulfur bacteria, they do not use H2O as hydrogen source. They use H2O, okay? They use H2S as hydrogen source. The oxidation product is sulfur and sulfur because they are not using water as a oxidizing source. They are using H2S. So the product, the byproduct will be different from oxygen. It will be sulfur or sulfate compound depending upon the organism and not O2. Hence, he inferred that O2 evolved by green plant comes from water, not from carbon dioxide. Earlier, what we used to think is this oxygen is coming from this carbon dioxide. So, by this study, we got to know that this H2O is releasing oxygen, because if we are supplying, for say, in case of green and sulfur bacteria, we are giving H2S in case instead of H2O. So this carbohydrate is getting formed, but here some sulfur compounds is getting released. If oxygen is coming from CO2, then here also the O2 should be released. But later on we found out that O2 is evolved by the green plant comes from water, not from carbon dioxide. He suggested this. But this was not accepted from all the people who were there. So later on, Rubin and Carmen, they proved that H2O is a source of release of O2 by this isotope, using this O18 heavy isotope. So what he did, he took one pot plant, he watered this plant with heavy isotope oxygen, okay, H2O18. So later on he found out that oxygen getting released by this plant is O18. That means whatever the Water we are giving, it is responsible for release of O2. Earlier, the equation we used to study was 6CO2 plus 6H2O in presence of sunlight and chlorophyll gives glucose plus oxygen. Okay, by this case we can understand, we can suppose that the CO2 is responsible for this oxygen, but later on the right new equation which is followed by all now is 6CO2 plus 12H2O in presence of sunlight and chlorophyll. It is forming glucose plus 6O2 is getting released, which is from this H2O molecule, and plus 6H2 is getting remain. This is a balanced equation. This is a correct equation which is followed now. Earlier, what we used to believe is this carbon dioxide is providing oxygen. What is getting evolved in the whole reaction, but this is not correct. So who proved that? Rubin and Carmen proved that. But previously, Von Neal has suggested that this O2 evolved by green plants come from H2O, not from carbon dioxide. Okay. So now comes the site of photosynthesis. The site of photosynthesis is nothing but chloroplast, which is a double membrane organelle. So how it looks? So it is the outer membrane. First membrane is the outer membrane. Let me highlight it. Okay. First is the outer membrane. This is the outer membrane. Next is this inner membrane. Okay. This is the circular DNA, because it is an autonomous organelle. That's why it is having circular DNA. Now comes this, they are having the 70S ribosome. Okay, this point you can see is the 70S ribosome responsible for protein synthesis. And this liquid which is in which this all the structure is being held is this stroma. Okay. Now imagine this green, wait a minute. This green stack you are seeing here. This is one thylakoid. Let me make it here. This is one thylakoid. This is second thylakoid, third thylakoid. You imagine as like you are putting so many coins one by one, like 10 coin, another one 10 rupee coin, another one 10 rupee coin. That one single 10 rupee coin is that what is our thylakoid. Okay. When we are stacking it together, then together they are known as granum, which is written here. Together it is known as granum. It is the stack of thylakoids. See, stack of thylakoid. Now, one grana is being connected to another grana by this structure which is called as stroma lamellae. This is stroma lamellae. One granum structure is connected to another granum structure by stroma lamellae. Okay. Now, whatever the portion of thylakoid which is in contact with stroma, this liquid, which is in contact with stroma is known as unuppressed region, which is in contact with stroma. Part of the thylakoid membrane, which is in contact with stroma, it is known as unuppressed. Whereas the part of thylakoid which is not in contact with stroma, because it is one above the other. So this middle part or central part is not in direct contact with stroma. So that is known as uppressed region. So why we have to understand this oppressed and unoppressed region, we'll get to know later on when we'll start the light reaction and a dark reaction, because these they have important role in that. So for now, you have to just remember that what is oppressed region and what is unoppressed region. Okay, let's move to the next. So this is what the site of photosynthesis is, that is chloroplast. It is a double membranous structure. The structure you have to remember because in this only the photosynthesis process is taking place. Okay, so let's move to the next slide. So this is how the reaction is. This light reaction is taking place in this. Wait a minute. Light reaction is taking place in this granum, that is stacks of thylakoids. Here the oxidation of H2O is taking place. Here the O2 is getting released. This is called grana, the stacks of sunlight. On the striking of the sunlight, there is a breakup of this H2O molecule, which is releasing electron and H plus. So O2 is getting released, and this ATP and NADPH2 is synthesized from this light reaction. It is just a overview. Okay, like I'll teach you everything in detail. So it is just an overview. So in the light reaction, what we are getting is this ATP and NADPH2. This ATP and NADPH2 is used in this dark reaction. What we understand through Calvin cycle, what happened in this Calvin cycle is CO2 from atmosphere will be taken, and this CO2 will be converted into sugar, and later on it can be converted into starch or whatever the things required by the cell. Now, this dark reaction will release ADP plus inorganic phosphate and NADP in the stroma. And from this, it is again getting used in light reaction to produce again this ATP and NADPH2. That is what the whole scenario which is happening in the chloroplast. Okay. So light reaction is taking place in this grana, and this dark reaction is taking place in stroma. Okay. Let's move to the next slide. So now comes the pigments. Photosynthetic pigments are special molecules, those absorb, transmit and reflect different colors of light from a visible spectrum of sunlight. What they do, they are the special molecules. Okay, they absorb, transmit and reflect different colors of light from a visible spectrum of sunlight. This visible spectrum lies from 300 to 700 nanometer. Okay, and this photosynthetically active radiation is from 400 to 700 nanometer. This is important. Pigment appears in the color which it reflect. For say, if a leaf is looking green, it means it is reflecting green and absorbing all other colors. When we are studying this Vibgyor, so what is happening is this green color is getting reflected, and all the other like violet, indigo, blue, yellow, orange, red, all are getting absorbed by this green leaf. That's why the color of the leaf is green. Okay. So now comes the photosynthetic pigments. So there are three kinds like chlorophylls, the most popular ones, carotenoids and phycobilins. Okay, let's move to the next. Now comes the chlorophyll. Chlorophyll is a green colored pigment. Important. Light is required for their synthesis. Okay. It is soluble in organic solvents. So there are different kinds of chlorophyll. First is chlorophyll A, most important. Chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E. Chlorophyll A is a universal pigment. Why we are saying universal? Because it is found in every photosynthesizing like organism, okay? Which is found in all O2 liberating photosynthetic organisms. It is blue-green in color. This color is very important. This chlorophyll A is blue-green in color. This color you have to remember. Chlorophyll B is accessory photosynthetic pigment. This chlorophyll is very important, and this chlorophyll B is an accessory photosynthetic pigment found in euglenoids, green algae, and higher plant. And the color of this chlorophyll B is yellowish green. This you have to remember. This chlorophyll is of yellowish green color. Yellowish green Okay. Next, structure of chlorophyll A. This chlorophyll A looks like a tadpole. Okay, this looks like a tadpole. What is tadpole? When there is development of a frog. Okay, the baby of frog once go through this tadpole like thing. That baby. Okay, when a frog is growing, then first it start with a tadpole. So this is the head and this is the tail part. So we are comparing it with the structure. Here we see this chlorophyll structure. This is the phytol rings part and it is appearing as head. And this chain, if we are writing it at vertical, it will look like a tail kind of. This is the head part and this part is looking as tail. So that's why we are saying that chlorophyll A looks like a tadpole. Okay. We are just comparing it with a tadpole. It is not a tadpole, but we are comparing. So there are four pyrrole rings. What are these pyrrole rings? This structure, one nitrogen containing this structure is one pyrrole ring. So such like that, there are four pyrrole rings. See, two, three, and four. So there are four pyrrole rings. So four nitrogen are there because each pyrrole ring is containing one nitrogen. Okay, here, here, and here. Four nitrogen are there. Central atom is magnesium. This atom is magnesium. And CH3 in side chain. In case of chlorophyll A and chlorophyll B, only one difference is there. The inside chain of chlorophyll A, CH3 is there. Whereas in case of chlorophyll B, CHO side chain is there. Just only the slight difference between chlorophyll A and chlorophyll B is this. So chlorophyll A is 55, H72O5 and 4 magnesium. This is the formula. And CH3 is in the second pyrrole ring. In the second pyrrole ring, this is the second pyrrole, it is having side chain methyl group. Here it is having aldehyde group. Okay. In chlorophyll B, it is C55, H70, O6, and 4 magnesium. Here, aldehyde group is in the second pyrrole ring. And there is a note that chlorophyll C lacks this phytol tail. Chlorophyll C does not have this phytol tail, what we see in chlorophyll A and chlorophyll B. So let's move to the next slide. Now comes the pigment that is carotenoid. Carotenoids are yellow to yellow orange in color, important. Pigments which occur alone inside the chromoplast occur along with chlorophylls inside chloroplast. Okay, these are the universal in occurrence, just like chlorophyll, except in new bacteria, and insoluble in water. This is important. They're insoluble in it. Chemically, they are terpenes and considered as most stable pigment. This is important. They are most stable pigments. Light is not necessary for their synthesis. In case of chlorophyll, we studied that light is required for their synthesis. Now let's divide the carotenoids. So carotenoids can be divided as carotenes and xanthophyll. So carotenes are carotenes. Okay, carotenes are yellow to reddish in color, yellow to reddish yellow, or we can say yellow orange. Okay, and there are three types like alpha carotene, beta carotene, and gamma carotene. In this, the beta carotene is very much important. Whatever the color we see in carrot, that orange color, it is due to this beta carotene. And beta carotene is a precursor of vitamin E. This is important. Now comes the xanthophyll. Xanthophyll is yellow in color. There are two types like fucoxanthin, diatoxanthin. Fucoxanthin is found in this brown algae. Okay. Now comes the functions of carotenoids. They are accessory pigments. They are not the main pigments as chlorophyll A. So they are accessory pigments. They make more photosynthesis more efficient by absorbing different wavelength of light. What is their work is just to absorb different wavelength of light and they protect chlorophyll A from photooxidation. If light will fall on chlorophyll A directly, so this chlorophyll A have chance of damage by photooxidation. So what they do, they protect this chlorophyll by photooxidation. This is how they are arranged. Okay, they protect chlorophyll A from photooxidation, and they also protect photosynthetic machinery by converting lethal nascent oxygen. Nascent oxygen is this free radical oxygen into unharmful molecular oxygen. They convert this nascent oxygen into unharmful molecular oxygen. So that's why they are known as shield pigments, because they are protecting chlorophyll A from photooxidation. They are they're converting this lethal nascent oxygen into unharmful molecular oxygen. That's why they are called as shield pigments. Beta carotene act as a precursor of vitamin A, as I told earlier, and they help in entomophily and zoochory. Why? Because they're having more bright color, bright orange, yellow, like this color. So they will give plant a very attractive color, which will help that plant to attract different insects. So this entomophily and zoochory, they help in that. So next comes the pigment phycobilins. They're hot water soluble pigment. They lack magnesium and phytol tail. They lack both magnesium and phytol tail. There are three types: phycocyanin, phycoerythrin, and allophycocyanin. So phycocyanin is blue in color. It is a copper containing pigment. Pigment. Whereas phycoerythrin is red in color. This is iron containing pigment. And next is allophycocyanin, which is light blue in color. They occur exclusively in blue-green algae and red algae as an accessory pigment. In blue-green algae, there's phycocyanin, and this phycoerythrin will be found in this red algae. So they are giving this red color. Okay. Now comes the reaction center. So this is how the chlorophyll and other pigments are arranged. So this is the chlorophyll A reaction center, what we call it as. It is the reaction center, main pigment of this light harvesting complex. And this is how the other pigments are arranged. Okay, around this chlorophyll, so that it is getting protected from photooxidation, and uh, they are helping in absorbing more, like, different kind of wavelength of light. So this is called light harvesting complex. These are made up of proteins and accessory pigments. So these together make this photosystem. This is also known as quantasomes. So there are two types of photosystem: photosystem 1 and photosystem 2. Photosystem 1 absorbs 700 nanometer wavelength light, whereas photosystem 2 absorbs 680 nanometer wavelength light. And this photosystem 1 and 2, these are numbered based on their discovery, not based on their work. Okay. This photosystem 1 was discovered earlier, so that's why they called it as photosystem 1. And photosystem 2 discovered later on, so that's why they called it as PS2. Okay. Next, absorption and action spectrum. Absorption spectrum, what it tells is the graphical presentation of absorption of different wavelength of light by a particular pigment. For say, this is the absorption, okay? So here it is 400 to 700 nanometer. Now the absorption here, this blue line is representing chlorophyll A. This is chlorophyll A. And this green graph is representing chlorophyll B. And this black one is representing this carotenoids. So what absorption spectrum tells us is the ability of pigment to absorb light of different wavelengths. So it was seen that chlorophyll A shows maximum absorption in blue light. How we are seeing it? Because see, the chlorophyll A is showing peak at this. This is how the Vibgyor is. Vibgyor. Okay. So in the blue region, it is having more absorption. Chlorophyll A shows maximum absorption at the blue light. It shows another absorption peak as red light. So here also the chlorophyll A is showing another peak at near to red light, and here near to blue light. So what we got through absorption spectrum is that chlorophyll absorbs maximum light in the this blue wavelength zone and red light zone. That is what the absorption spectrum tells us. Now comes the action spectrum, the graphic curve depicting the relative rates of photosynthesis at different wavelengths. So this is 400 nanometer wavelength. This is 700 nanometer wavelength. So here it is found that photosynthesis is taking place more in this blue zone and in this red zone. So if we are overlapping these two, the absorption spectra and the action spectra, so what we found is this graph. This is the overlapped version. Okay. So now 400 nanometer to 700 nanometer. Now it is seen that chlorophyll, where the chlorophyll is getting a peak, in that area, this action spectrum is also getting a peak. Whereas the chlorophyll A is getting peak at red zone, and here also this absorption and this action spectrum is also getting a peak. So what it says, what we concluded through it is, it shows the wavelength at which there is a maximum absorption of chlorophyll A, that what we studied in absorption spectra, that is in the blue and the red region. It also shows higher rate of photosynthesis in that region. So what we conclude is, hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis, because wherever the chlorophyll is getting more absorption of light, there we are getting more higher rate of photosynthesis. So by this graph, we can conclude that the chlorophyll A is the chief pigment associated with photosynthesis. Next, let's start with the questions. First is oxygen, which is liberated during photosynthesis, comes from carbon dioxide, water, chlorophyll, phosphoglyceric acid. So it comes from water. How this H2O in presence of sunlight converted into 2H plus electron plus half O2. This O2 is getting released. So this oxygen is coming from water. Next, the law of limiting factor for photosynthesis was given by Robert Hill, Krebs, Calvinia, Blackman. So it is Blackman. This you have to remember. Law of limiting factor for photosynthesis. Next, the wavelength of light responsible for Emerson's enhancement effect. So see, what we studied is this 400 nanometer, it is 700 nanometer. So we are getting one peak here, then again one peak here in the action spectrum. And here at 700, this is 700, okay. At 700, there is sudden dip, and this dip is known as red drop. This is one concept, red drop effect, because photosynthesis rate is suddenly getting dropped here at 700 nanometer. Beyond that, there will be no photosynthesis taking place. Second concept is Emerson's enhancement effect. When we are providing red wavelength plus far red wavelength together to a plant, to a green plant, then the rate of photosynthesis, what we see is maximum. When we are providing red and far red wavelength, then we saw that the rate of photosynthesis is maximum. Why? Because When red and far red is being provided, then PS1 and PS2 both are working very hard because this red is getting absorbed and this far red is also efficiently absorbed by these two, PS1 and PS2. That's why we are getting maximum rate of photosynthesis, and this is called emerson's enhancement effect. So wavelength of light responsible for emerson's enhancement effect is this, both 600 nanometer and 700 nanometer. This is important, this red wavelength and far red wavelength. 680 and 680 nanometer and 700 nanometer, they are getting both. So that's why this PS2 and PS1 are performing very well. So that's why we are getting the rate of photosynthesis maximum. That's it, what emerson's enhancement effect. These two concepts are there, this emerson's enhancement effect and red drop effect. Very important. Next is the name of the scientist who first pointed out by bell jar experiment that plants purify foul air. We studied recently this bell jar experiment. Who was the scientist? This was the Joseph Priestley. So you can join this online master course, which is there available on the AgriAddict website. You can go through all the courses. We are currently conducting this agronomy mock boost for ICR PG 2025 examination. Many tests have been already uploaded. Some will be uploaded soon. So you can take benefit of that. And there are so many videos available for you. So you can go through all the videos, and class notes are also there. You can see here. So just be in like touch with the AgriAddict YouTube channel also. We will be uploading many lectures on it. And this is the website. You can go and take benefit and can kick boost your preparation. Thank you.